Okay, so today I'm going to be talking about the convolutional neural network-based image recognition code, faster RCNN.
Mhm.
So as a bit of an overview, faster RCNN is essentially the third iteration of a code called RCNN. So it's a object detection convolutional neural network code. This was initially published for the first time in 2015, and it was an update to fast RCNN, which is an update to RCNN. The key differentiating factor between RCNN and fast RCNN is the implementation of something called the region proposal network into the region selection process. So this was done in order to mitigate the problems of the previous algorithm called selective search. I'm going to talk about this in a lot more detail later. This is just a bit of a high-level overview. So in comparison to the previous iteration, fast RCNN, it was found that faster RCNN was able to speed up computational time by 11 and a half times, which is incredibly significant. It's fantastic. Some people say that faster RCNN is fast enough for use in real-time object detection. This can be debated, but it's definitely at the very least pseudo real-time image detection. On the right here, this is showing a graph of the decrease in computation time as you go from RCNN to fast RCNN to faster RCNN. And the image above is showing what a bit of an output from this image detection looks like. You have all these different objects in the image. They are all boxed to separate them from the background, and they are identified. So to talk about the origin of this RCNN, it's known as region-based convolutional neural network, hence RCNN. It was a code published in 2014 by a UC Berkeley lab. This code is essentially revolutionary in terms of image recognition goes. This was the first time that a region selection proposal was actually coupled with a convolutional neural network. Previously, you could do object detection, and then you might have to manually identify the bounding box with each object. This was the first instance where stuff was ever properly coupled. The way that regions are selected in RCNN is through an algorithm called selective search. I'll discuss that a little bit further. But basically, each region that is selected with selective search is passed through a CNN. They used AlexNet in this case. It's transformed into a feature vector. And then this feature vector is passed through an SVM, a support vector machine, and that's what tells you what this feature is, and it's within what box. SVN is also a convolutional neural network, and that's why it's able to be used for feature detection. So on the bottom here, you have a bit of an architecture of how the whole thing works. You have an image, bounding boxes are selected, features are extracted with the network, they're classified with the SVM, and then a final fourth, somewhat optional step is used called the bounding box regressor. This is what improves kind of the bounding box that isolates your image. So going from RCNN to fast RCNN, one of the biggest problems with RCNN, sure, it was absolutely fantastic, but it definitely had a few deficiencies. The whole thing wasn't properly coupled. It used two isolated neural networks, one for object detection and one for the bounding box algorithm. So this slowed stuff down a lot, and it also resulted in a lot of computational problems because you had to cache all the results of the model on your local machine. So the storage use would be literally hundreds of gigabytes. Fast RCNN rectified this problem by introducing a fully coupled neural network. So basically, the regions of interest are selected through selective search once again. They are passed through the convolutional neural network to decompose them into a feature map. This feature map is then used as an input into the region proposal feature map. And basically, the goal of this is you're using one central neural network actuated in two instances to both detect your features and detect the boxes around them. This was an absolutely colossal upgrade from RCNN in terms of computation time. It was on the order of 9 times faster. But again, it's still a little computationally inefficient due to the selective search algorithm. So yeah, now the fun part, the transition from fast RCNN to faster RCNN. So fast RCNN is fantastic because you're finally coupling only a single neural network for the entire process. But the problem is that you have two sets of calculations that cannot be computed in parallel. You have the feature detection and then you have the feature classification. Those just had to happen in series with fast RCNN, and that introduced a lot of computational slowdown. So as you can see on the bottom here, this is a graph comparing the training time on the left from RCNN to fast RCNN. So that's where you see a fantastic improvement. On the right is the test time. So when you're actually using real data trying to use the neural network, the region proposal time is clearly occupying a significant slot compared to the computation time. So the key problem here is the use of the selective search algorithm. That's kind of at the core of why you can't parallelize the two neural network processes. So to get around this, faster RCNN scrapped the selective search algorithm and it imported something called the region proposal network. The RPN is basically a way to parallelize the computation here. So the architecture is you take an image, you pass it through your convolutional layers to isolate the features in the form of a feature map. And then at the same time, from that feature map, you're using a trained neural network to isolate region proposal boxes, as well as having the features. Those are passed into the next layer called the region of interest pooling layer. And that is where your regions of interest are basically applied to the features, as well as the feature classification process. So a little bit more on the RPN here. Let's see. Yeah, this kind of goes over a little bit of what I just talked about. But basically, within the region proposal network, the RPN, you have different sorts of kind of feature sizes that can be detected. And that's how it works to iteratively refine the bounding box. And once you've, the big attraction to having the neural network coupled here is that as you're progressively successfully identifying accurate bounding boxes, that minimization of error is propagated through the whole model, and that works to continually improve it. So that's one of the big reasons that the RPN is superior to the use of the selective search algorithm. So once again, here's kind of a summary of code developments over time. You have RCNN, which was fantastic, but it had the problem that it used two discrete neural networks that were not properly coupled. You have fast RCNN, which changed the feature map. Initial feature map. Yeah. So you have the use of a single coupled neural network, but it has to be actuated in a series of steps. And then faster RCNN scraps the selective search algorithm, which was kind of the core problem with fast RCNN. And through the use of region proposal network, you parallelize all the computing and you significantly accelerate the code. So a case study talking about applications of faster RCNN in biological imaging. So what this study did was it used computer vision to analyze like microscope images of cells and isolate what were different types of malaria cells versus like assorted immune system cells like red blood cells and white blood cells. So the study started by obtaining a fairly comprehensive database, those 1,300 microscope images with about 100,000 cells. The extremely time-intensive operation here, they had a few experts from their lab comb through the entire database and isolate and categorize different cells. So you can see on the image on the left here, you have the different blue boxes. Those blue boxes are indicating what are a red blood cell, which is something like 80% of all the cells obtained in the samples. And then you have the orange and yellow and purple and green. Those are indicating different actually interesting categories. So that's what the model's working to isolate. So in order to do a good quantitative study, because the goal here was it wasn't so much to like go through all of these images and actually identify malaria cells. It was more to create this neural network as a tool for future research. Because presently, convolutional neural networks for image analysis have been frequently used for biological imaging, but one of the big problems is that most of the time the information remains proprietary and it never actually gets published as like a functional tool for other labs to use. This study really wanted to change that by actually creating a useful implementation of computer vision for future research. So for the benchmarking, they had the Sorry, it's a Friday at 4 p.m. So for the benchmarking exercise here, they used a conventional tool, which still uses a convolutional neural network, but it's not known to be necessarily the strongest. It's a program known as Cell Profiler. So that's what obtained their like benchmark data. So saying like how good are these new tools compared to what we usually use. The way they implemented a faster RCNN in this study is they did it through a two-stage process. They had faster RCNN basically isolate the images into binary. Is this a red blood cell or is this not a red blood cell? That was the first step. And then what they actually did is they implemented a second CNN using AlexNet to further classify. So like a fine grain image analysis being like, we know these are all not right. We know these are all not red blood cells, but we don't actually know what they are. We're going to use AlexNet to determine exactly what they are. So this dual stage system is what they used for the image analysis. They did do benchmarking on both the single stage and the dual stage versus the standard benchmark though. On the bottom here is, I won't go too deep into this, but it's talking about the different levels of convolution they do in the images. So that's how they're doing different levels of feature extraction. You see the standard faster RCNN architecture here, where following the convolution of the image to extract the feature map, things are passed through the region proposal network, and you finally result in a object classification and region isolation step. So as far as the results go, they are quite impressive. On the left here, this is the cell profile or baseline. So you can see on the bottom here, the accuracy of Cell Profiler, which is like your standard way of doing it, is only 50% when compared to the human done like actual real results here. In the middle, you can see the single stage faster RCNN implementation. You have a slight improvement on the benchmark, but it's still not all that significant. But on the right here, this is a confusion matrix. So the confusion matrix is on the middle column here, sorry, on the middle diagonal, that's what's showing when both your model and your actual result match up. And then the off diagonal entries, these are showing where your different errors. So like, what are your false positives in both models? You can see here that the entries are they're almost none. This reflects the accuracy of 98% in the two-stage faster RCNN and AlexNet implementation. So this is truly fantastic. This is almost twice as accurate as the most conventional method of doing this. Results discussion. So one other way of isolating cells from other cells is to use basically experts who are extremely good at this. So to do that in this study, they took two experts from different labs who were experts in malaria cells. Oops. On the right here, you can see the average accuracy. So the average accuracy of 72% it's quite good, especially compared to the baseline cell profiler method, which has only 50% accuracy. But the problem is doing a human-based analysis is extremely time intensive, and it's not really the best way of doing things, hence why these software tools exist. So you can see here that using the two-stage faster RCNN and AlexNet implementation, you significantly improve on the average from even like a human vision-based perspective. It's fantastic. Concluding remarks. So this presentation served to kind of go over how faster RCNN developed as a result of the initial RCNN code following fast RCNN code, and then finally faster RCNN code. This has resulted in the development of a pseudo real-time. So it's fast. It's not perfectly fast, but it's quite fast. High accuracy object detection classification code. Initial proof of concept studies in medical imaging demonstrate how this can be an extremely valuable tool for image analysis, being both more accurate and faster than conventional approaches and like human eye-based approaches. And the last remark is that as good as faster RCNN is now, it will probably be rendered obsolete within quite a fast time span, because all computer vision codes, these the development, the speed at which they develop is quite staggering. So one possible alternative to faster RCNN is actually the YOLO imaging code. But it's hard to say that YOLO is necessarily better than faster RCNN. YOLO is most certainly faster, but it doesn't necessarily give as good of a fine-grained detection response. So Some would argue that YOLO is the next step from faster RCNN, but in my personal opinion, I don't think it is. It's simply a different tool. That concludes my presentation. Thanks.
Okay, good. All right. So having said that, do you have any idea of YOLO?
Like a decent, I have a functional knowledge of it.
Okay. So I would, okay, so you presented this interesting case where, they applied LXNet and then they applied faster RCNN.
It was faster RCNN and then AlexNet.
Faster RCNN and then LXNet. So can we go back to that slide for a moment?
Yes.
Yes. So they applied faster RCNN. So in the faster RCNN, they are obtaining the different kind of, here, these are blood cells. Is that right?
Generally just cells. You have cells, then you have virus cells and bacteria cells.
Okay, so they are trying to separate between two kind of cells. They're doing this kind of classification using faster RCNN.
Correct.
But then why are they also using LXNet? Because if by faster RCNN, you're able to classify, and faster RCNN should also be able to produce these bounding boxes, which I don't see, because do you understand what is LXNet?
I vaguely understand AlexNet. I don't have a huge understanding. So I'll show you here. It's easier to see when I zoom in. In faster RCNN, they're still doing the object detection and object classification thing here. But what they're doing is they're only doing kind of a binary, I won't call it a binary search, but a binary classification saying, is this a red blood cell or is this not a red blood cell? Because the red blood cells make up almost the entire image. So once they've isolated what the red blood cells are or aren't, that's when they pass it into AlexNet. And this is where they're doing the fine grain classification of among all the cells that are not red blood cells, what are all these different cells?
Do you understand like why would they couple it? Because faster RCNN individually should be able to just, classify what are red blood cells and what are,
Yeah, it's a good question. So the general answer from the paper is something about because they only have a limited amount of training data for cells that are not red blood cells, like they have a significant amount of data to like isolate what is or isn't a red blood cell, but following that, actually characterizing what the different cells are, it was they didn't explain it super well why they did this, but the use of AlexNet, they had a sufficient database that they could uh, basically get better results that way. I didn't have a great understanding of why they did it though.
Yeah, because what is LXNet? LXNet is just a series of CNN. So it's just series of convolutional layers that
Yeah.
would finally give you, results. So it is very same thing that you have in the backbone, the backbone that you just showed in the diagram.
Yep.
But it has just more layers to it.
That's what like I think I understood it to be. Yeah, it's just another CNN. It's just on a different architecture.
Yeah, because normally the backbone of faster RCNN is already inspired by AlexNet, VGG, or by GoogleNet, or by different things. So just applying it 2 times, it just seems, to me, a little bit not well understood, because even because these YOLO or faster RCNN, they are quite robust in doing analysis for most part. So if there was a shortage of data, I would prefer like techniques like data augmentation.
Yep, data augmentation was done in this data set as well to try and improve the data set for the non-red blood cell cell types.
And if we apply this faster RCNN to our bubbles data, what do you think? Would you You think that we would do the similar approach applying through AlexNet and faster RCNN, or it could be directly applied?
I
Is there a
think
constraint of
I don't see why the second step would be necessary, because as demonstrated here, faster RCNN did an excellent job of like identifying kind of a binary yes/no criteria. And when you're analyzing bubbles, it really is a binary. Is this a bubble or is this not a bubble? So if we're treating it kind of analogously, I think that just applying faster RCNN for bubble identification would be more than sufficient, because it's very good at that.
Yeah, so you would directly apply, right?
Absolutely, yeah.
Okay. Now you use the term selective search algorithm multiple times.
Yeah.
Now, but I never understood what is selective search algorithm, because it was replaced by RPN.
Yeah.
So I think in faster RCNN, they replaced it by RPN.
Yep.
So what is the classic difference between selective search and RPN?
To my best understanding, selective search isn't a convolutional neural network based like bounding box algorithm. It's so it doesn't necessarily learn from like successful iterations. It more, I believe it breaks the image down into like 2,000 different possible bounding boxes, and then it
Based
does
on
bounding.
what? Based on what? Like there has to be some criteria, that it is taking, is it, there are different kind of things. Is it doing by, because it sees a different edge, it sees a different color intensity, it sees, because if you Google the selective search algorithm, they show the image and then they broke down that image into different patches of colors. That's a common image that you see. So like if I give you the image, any image, what do you think is the outcome of selective search?
Yeah, so it definitely does some level of image analysis, but it's decoupled from the main convolutional neural network that the rest of the process operates on. So it doesn't have like that learning step where following bounding box regression at the end of it, it doesn't make those corrections to further improve the model. That's my kind of working understanding of it.
So it's just something that doesn't improve. It just is the initial, like you're breaking your image into multiple small parts, and that's getting trained, right? And it's not improving
Exactly.
the initial image, right?
Exactly. Because at the end of all this stuff, you always have that bounding box regression step where the bounding box is improved to better isolate the feature. In selective search, that bounding box regression never actually reaches the selective search algorithm. So these iterations, they don't actually improve it. That's why it's kind of computationally heavy, I believe.
Okay. And similar thing applied to SVM. You said that it's also CNN based. SVM is CNN based. When you were explaining,
Yep.
Here,
scrolling up somewhere.
The next slide, it's a very good comparison that you have provided between faster RCNN and the final slide. Yeah, this is good too. Yeah. So in RCNN, we use SVM.
Correct.
So any idea? So, this is very classic things that I did, the activity in the class that, you have an image, it extract the features. After the features, it is going to a fully connected layer.
Yep.
And then after a fully connected layer, the things we did in the class, the fully connected layer composed of neurons. So ultimately an image is passed, you apply, all these filters and max pooling, and it is converting a very high resolution image into, into very low resolution where, the 5 pixel information is comprised in one, and it's just to make the computation easy. And then that particular last image, it's getting flattened in the neurons. You flat the image and you apply full connected layer, which is just, the 2 horizontal line that you see in your thing.
Yep.
I think this is written FCN, fully connected, right there. So the image is flattened, and now it's goes under that neuron treatment. It goes under the neuron, and it predicts. It directly predicts the classes that it has to predict.
Yeah.
Then they are using another same algorithm, that is SVM. When we have already predicted classes here.
Not exactly. Um, so in RCNN, the big reason that like the big problem with it is that you're using AlexNet. Well, in this case, pretty often you use AlexNet, and that's what's doing feature identification from an image. So basically being like, here's a thing, here's a thing, here's a thing, and that operates as one isolated CNN, and the output of that are your feature vectors, but it doesn't tell you what those feature vectors are. Those feature vectors are now passed into the support vector machine, the SVM, and the SVM is what's actually used to classify those feature vectors. So they operate in like an isolated sense, and there's no communication between the two neural networks aside from passing like the vectors.
So this looks, I think the understanding is clear. What you speak, whatever you are saying right now. But my point is that when I see this fully connected layer, so there is a thing. There are two things into these networks. One of them are convolutional layers. Other are fully connected layer. And once you have convolution layer, into that, you are simply extracting features, or you're simply creating a low resolution image by using convolution layer. So basically you have a matrix of numbers that gets multiplied with your image, and you get this. But then you're passing it to fully connected layers. And if you pass to the fully connected layer, there you apply all kind of probability kind of functions, which is like cross entropy, binary cross entropy, softmax. And all these probability functions, they actually try to, find the nearest neighbor with the trained data and try to finally predict this is, what kind of feature. So this is typically happening in FCN. So I'm simply looking at two classification, two classification segments into one model. One classification segment is FCN, and other is SVM. So there's a double classification happening here.
That's correct. The first step is basically isolating features from background. And the second step is telling you what those features are, and they are not connected.
Yeah. Right. Yeah. So if they're not connected, then it makes sense.
Yeah. Oh yeah. They're not. That's like why our CNN is a little strange, and that's why it's so slow, because you literally have to train two completely discrete neural networks that the only thing that those networks do is pass a vector into the next one, but there's no communication aside from that.
I think now it makes sense.
Yeah.
Because I clearly see like there are two classification layer, and that confuses me, like why you need it.
The answer is it's kind of weird, but these people were the first people to do this, so it was never going to be perfect.
It was, no, that's correct. Okay, this is the final question. We go to the comparison slide of RCNN, fast RCNN, and faster RCNN.
This one here.
Yeah. Okay, so yeah. So we know RCNN, they removed, SVM, and then they added something, called RPN.
So the sort of initial feature map. Oop, there's a typo here. But basically, the difference was in RCNN, you have the two neural networks. In fast RCNN, it turns into one neural network where first the image is broken down into a feature map. So like isolating what all these features are. And then it goes into the next step where from the feature map, that's where you're applying a classification and bounding box scheme. So it's two steps.
Right.
And that's why it's all operating in a linear manner here.
And so RCN to fast RCNN, and then to faster RCNN. So like this is looks very attractive to just see that in RCNN they had two different branches as we discussed for classification.
Yep.
In fast RCNN, they're just coupling everything into one operation, right? And
Essentially, yeah.
yeah, so they're just passing it through once. They are passing through your convolution layer where they're extracting the features.
Mm-hmm.
And then these features, they're going through separate branches. They are going through, one is they would just predict what class it is. That is using a softmax function, which is your probability function. And other
Mm-hmm.
is, they are trying to regress the bounding box.
Exactly.
Now, coming to faster RCNN, now what is happening in faster RCNN is that like the bigger blob that you have as a pyramid. So here, in the last image on the screen, you have this orange color, like a big blob.
Yep.
Do you know what it is, this big blob here?
Yeah, so fast RCNN works quite similar to fast RCNN, where the image is passed through all your convolutional layers, and that's how it's transformed into a feature map. But the key thing is that instead of doing like a split where one step is feature classification and one step is like bounding box regression, they all happen in parallel at the same time from that feature map. So that big orange blob is the convolutional step transforming the image from like an image into a feature map.
Right. And then RPN. So this is the only term that's, doesn't like, so what RPN is doing here? So RPN is trying to, it's a pyramid network, right? It's a pooling network.
What was the first thing you said? It's a
RPN pyramid.
That's a good question. I believe it's a pooling network.
RPN and ROIP.
That is region of interest pooling.
Yeah, the changes is removal of SS in favor of RPN.
Removal of selective search in favor of RPN. So it's using the informed convolutional neural network scheme to do region of interest proposals.
Right. Yeah. So here just this is kind of a little bit not clear, like RPN and like what is happening after feature is extracted. Okay, you converted an image to a low resolution image. Okay, done. Then what is happening in RPN?
The RPN is your region proposal network. So that's what's looking at your entire feature image, and it's proposing it's like proposing the bounding boxes for being like, this is a feature, this is a feature. So it's the like region of interest proposal process.
Then it is going to ROIP.
Yes, that's region of interest pooling, and that's where you have a combination of features as well as their bounding boxes.
Now it makes sense because it's a region proposal network. So everything that is extracted in a very low resolution image, now you are trying to propose that it's a cat, it's a horse, or it's a dog in the
Here,
image, and you create
this is a better example of it on the right here. So you have the convolutional layer where it's transformed into a feature map, and then you can see at the same time that your feature map just passes straight to the region of interest pooling, but at the same time, you're having your RPN generate your region proposal maps, and that's what's isolating features in the feature map. And from there, it goes to the final classifier.
Right, right. And when and this is ROI pooling, and in ROI pooling, they are trying to, again, condensed all the features into further,
Exactly, yeah.
It's good. I mean, you have developed quite understanding with these papers. That's impressive.
I had a lot of fun. It was really cool. And I want to figure out how I can use this in my own research, honestly. I don't think I can, but I'm trying to implement.
Yeah, I mean, for bubbles and everything, I would be very happy, that, if you, because the point is that you are not really a microstructural or a TEM person. So, it might not be of much interest to you in terms of image analysis, but
it's still the same statistics if you're doing like statistical analysis with machine learning. but it's not image analysis, but a lot of it's the similar math. So I think I'm probably going to try and do ML based. So I do a lot of sensitivity analysis. So like when you look at the output of a model versus the input, if you're doing inverse uncertainty quantification, which is a lot of what I do, you're kind of determining from your results what are like model sensitivities and how those can impact your results. And that's a big place where I'm trying to, once I learn more, actually use machine learning concepts. I don't think I'm ever going to be doing image analysis with it, which is kind of sad because it's really cool and I like it, but I can still use a lot of the same principles.
Yeah, basically the principle of backpropagation,
Exactly.
where you're trying to optimize your input based on the output. Yeah. Mm-hmm.
And then there is a small chance, so I'm somewhat involved with a research project that's doing like experimental work on fee crowd cladding, and they're doing DICs, that's digital. Wow, I forget what DIC stands for. It's been a while. I just noticed DIC. You know DIC, right?
Yeah, I think it's
Yeah.
yeah. It's
Either way,
more digital. Yeah, I'm also it's skipping my mind too.
image correlation, digital image
Yeah,
correlation.
digital
Yeah.
image correlation.
So we have like an absurd data set that we're continuing to grow. And I think that a very cool opportunity to apply computer vision would be to apply that for DIC, because one of the limits in DIC is at like super big strain rates, you start to have your actual like speckles start to rip. And when your speckles tear, your like conventional software, it can't do it anymore.
Okay.
And when we're doing the DIC for this, we're doing clad bursting. So we're actually like heating and pressurizing cladding tubes until they literally blow up. So the strain rates are massive.
Okay.
So I'm interested if computer vision can be applied there to
Maybe
possibly improve it.
yeah, if you do the live video, you have some videos, and if you have cameras attached, you can really process that.
Exactly. And I have that data. I have terabytes and terabytes of it.
The videos that gets, that would be super cool,
Oh,
I would
yeah.
love to help you, if something like that.
Oh, that'd be fantastic. Once I have a little bit of time, when I wrap up coursework and my other research stuff, I want to really put some time into it because I think it could be really cool.
Yeah. It appears cool, if you have this terabytes of videos, to just see where that, the how do the fracture starts to happen. It would
Exactly.
be
Yeah.
super cool. Right. I think good understanding, Jonathan. It was nice.
Yeah. Thank you. Thank you. It was fun.
Yeah.
All right. Take care.